Bem-vindos ao nosso espaço, um mergulho profundo nos assuntos mais interessantes de esportes, ciências e temas gerais do momento. Aqui você vai encontrar conteúdo de qualidade, com análises empolgantes e descobertas fascinantes que vão expandir seus horizontes. Nossa missão é trazer informação relevante de forma dinâmica e descontraída. Queremos que você faça parte dessa jornada incrível de conhecimento e diversão. Se gostar do nosso conteúdo, não esqueça de deixar seu like e se inscrever no canal para não perder nenhuma novidade. Ative o sininho também. Aproveite!
If you love science, don't forget to like and subscribe. For decades, scientists have grappled with the physical reality of a memory. We know how to form them. Neurons fire, synapses strengthen, and new proteins are synthesized. But the biggest mystery isn't how we form a memory, but where we keep it. Where is the substrate of your deepest, most enduring recollections? The name of your first pet, or the smell of your childhood home? The answer might be in a molecule we traditionally treat only as a humble messenger, RNA. We call this the Ribonucleic Scaffolding Paradox. It challenges the standard model of memory, which holds that long-term storage relies on synaptic plasticity, the physical strengthening of connections between neurons. When you learn something new, the synapse remodels itself, often by creating new proteins directed by messenger RNA, mRNA. Think of it as building a stronger bridge every time you cross it. But this model faces a critical stability problem. Synaptic proteins are constantly turning over, degrading, and being replaced. The physical components of that strong bridge are gone within days or weeks. If the proteins defining the strength of a synapse are temporary, how can memories last 50, 60, or 80 years? How do we maintain structural integrity against perpetual molecular decay? This is where the paradox forces us to look beyond the sequence and into the structure. Researchers are now proposing that the truly permanent archive of memory is not the sequence of DNA, nor the transient protein products, but the three-dimensional, stable scaffolding of specific non-coding RNA molecules, or ncRNA. RNA isn't just a linear code, it's a master folder. It twists, loops, and self-assembles into complex shapes, structures often called pseudoknots or riboswitches. These structures are profoundly stable, far more resistant to decay and molecular turnover than the proteins they might help produce. The scaffolding hypothesis suggests that a long-term memory is encoded not by the asterisk message that RNA carries, its sequence, but by the highly specific, persistent shape it adopts. Imagine a complex piece of origami. The paper is the RNA sequence, but the final folded sculpture, the crane, the boat, is the memory state. You can replace the underlying paper many times, but as long as the complex folds are perfectly replicated in the new material, the information, the structural state, persists. That is the essence of structural memory. How does this happen in the neuron? When a long-term memory is consolidated, specific signaling pathways trigger the synthesis of certain ncRNAs. These molecules immediately fold into unique, intricate configurations. Crucially, these unique structures act like molecular templates or scaffolds, organizing surrounding proteins and enzymes into the precise spatial arrangement required to maintain the long-term potential of that synapse, regardless of protein replacement. Furthermore, these structural RNAs possess catalytic properties and can sometimes maintain their shape even if a small part of their sequence is damaged, exhibiting a robustness that simple protein interactions lack. They function as a hard drive, where the information is stored geometrically. This geometric state can be replicated across generations of replacement molecules, providing the necessary molecular permanence that synaptic plasticity alone cannot explain. One particularly compelling avenue of research involves small inhibitory RNAs, siRNAs, and pRNAs, which are known to influence gene expression by regulating DNA methylation and chromatin structure. If a specific ncRNA fold can permanently alter the accessibility of genes related to synaptic growth, it has achieved an epigenetic memory state, a lasting biological instruction set, secured in its structural integrity. This paradigm shift changes how we view neural computation. It means that the memory capacity of the brain is not limited merely by the number of synapses, but by the potential number of stable, complex folding configurations that these RNA molecules can adopt. The brain is utilizing a biochemical version of topological storage, where complexity is key to resilience. If the RNA scaffolding paradox holds true, future treatments for neurodegenerative diseases like Alzheimer's wouldn't focus solely on rescuing synapses or cleaning up protein plaques. Instead, they would target the stabilization or repair of these crucial RNA molecular folds, effectively restoring the foundational architecture of archived recollection. It's a compelling vision, our deepest memories residing not in transient chemicals, but in the durable, exquisite geometry of life's most versatile molecule. For more incredible facts like this, don't forget to subscribe and turn on notifications.